We represent the part of the meta-analysis group that is currently just about to submit the data for the final meta-analysis, and that's the NEAT trial data, and I'm going to just present some data to you from that today. But just a word about what Angelo said about HER2 and TOPO2 as predictive biomarkers in this context. The association between HER2 and anthracycline response was really accidentally stumbled upon when people were looking for prognostic studies of HER2. And we then saw an association between HER2 and anthracycline response, which was not widely substantiated, and Angelo's given you strong evidence of that. Now, we know HER2 is in a region of genes which is frequently co-amplified, and we then scanned that gene region, and we found that there was another gene, TOPO2, that had a very attractive profile as a potential predictor of anthracycline response. Some in vitro data suggested that amplification of TOPO2 was associated with sensitivity to anthracyclines, but critically, deletion was associated in vitro with resistance, and that's one of the things we must recall when we interpret data on deletions of TOPO2 in the clinical setting. We should also remember that there are a whole load of other genes in this amplicon which have roles in breast cancer. And indeed, the chromosome on which they sit, chromosome 17, is the second most gene-dense chromosome in the human genome. It contains other breast cancer genes which are associated with DNA repair, particularly BRCA1, a gene we all well know is associated with high risk of familial breast cancer. And other genes on the other arm of the chromosome, which include P53, which is a functional gene critical in response to chemotherapy. So what we sought to do when we entered this study was to look in a large clinical trial and address the hypotheses that, yes, potentially the markers which were being discussed, HER2 and its signaling partners HER1 and HER3, might drive anthracycline sensitivity, as might TOPO2, and to test that prospectively, but also to explore that chromosome 17, polysomy, alteration of the chromosome itself, might predict for anthracycline response. And in fact, that HER2 and TOPO2 might be surrogates for this polysomy 17. And that was an idea that came to us last year and has been introduced into the study. This is the NEAT trial, which was published in the UK by Professor Poole, who's in the audience if you wish to discuss it further with him. It's an anthracycline followed by CMF study versus CMF alone. It has two arms, which are a Scottish arm called BR9601, the data that Angelo referred to as a Scottish study in the meta-analysis, and the larger NEAT study containing 2,000-odd patients. And this provided us with a unique opportunity to train the markers that we're looking at in the BR9601 study and then validate such markers in the larger NEAT study. So we have a two-step approach that we can take. Unfortunately, when we did the treatment by marker analysis in the NEAT trial, and this is data on just short of 1,650 patients, for all the markers which Angelo has talked about, none of them showed a predictive interaction with anthracyclines. So HER2 positivity by amplification, TOPO2 amplifications or deletions. As discussed by Angelo, there was no significant treatment by marker interaction suggesting that these markers could be used to select patients who would benefit from anthracycline therapy. And insofar, our results are entirely consistent with those presented by Angelo De Leo. Moving on to the chromosome 17 polysomy as a predictive biomarker, well, it is a simple marker. It just involves duplication of the entire chromosome, we believe, and therefore allows for duplication of alteration of multiple genes without amplification. But actually, it's also associated with a phenotype known as chromosomal instability, and chromosomal instability has been associated with anthracycline sensitivity in vitro. It's detected by FISH. In fact, if you're doing a test for HER2 gene by FISH to determine whether a patient gets Herceptin, this comes free. This is the test against which the HER2 gene ratio is controlled. So we're in a very good position because it's a very pragmatic marker that could be rolled out very quickly. And we performed two studies. One is the pilot study looking at BR9601, the study that I mentioned earlier, but also at the Canadian MA5 study, which Angelo also referred to, because it showed contradictory results with regard to HER2 to our own study. And then we took forward a confirmatory analysis in the NEAT study. So first I'll show you the data from a poster that will be presented on Sunday morning, poster 6059, which shows the interaction between chromosome 17 and polysomy and response to anthracyclines in a combined analysis of the BR9601 and MA5 Canadian trials. And here the test for interaction is significant, with a hazard ratio of 0.63 for patients who are polysomic to benefit from anthracyclines. That's a 37% reduction in risk. And as you can see in the two sub-analyses, in fact, the polysomic group has almost a 40% reduction in risk if they're given anthracyclines, whereas there is no strong evidence for a benefit in those patients who have no increase in copy number of chromosome 17, shown in the left panel. And this is then the similar analysis performed in the NEAT study, and the full data will be presented this afternoon. And this confirms the test for interaction at the hazard ratio of 0.59 with a p-value of p04. Again, the polysomic group showing a significant benefit from anthracyclines, and the normosomic group showing no benefit apparent from anthracyclines. What about the possibility then that the previous results we have seen have been because HER2 and TOPO2 are strongly associated with polysomy of 17? Well, this is a forest plot. It's a bit difficult to take you through, but the top line is essentially those cases who have no alteration in HER2 and no alteration in chromosome 17. The next line are HER2 amplified patients who have no alteration in chromosome 17. The next line are patients with polysomy of 17 but no alteration of HER2, and the final is both altered. And what you can see is that the two groups most to the left of the line suggesting that anthracyclines benefit are the polysomic groups, irrespective of whether they have an amplification of HER2 or not. So there are HER2 negative patients who are benefiting from anthracyclines based on this subgroup analysis. There is a slight trend that there might be an effect of HER2 amplification, but it's certainly significantly weaker than the two subgroups with polysomy. And the bottom panel is essentially just a repeat of that, looking at TOPO2 alterations. And again, it's the bottom two groups that lie to the left of the line, that is polysomic groups with or without TOPO2 alterations, and the group without polysomy that has TOPO2 alterations lines close to the line of unity. So in the NEAT trial, which will be combined, as Angelo said, with his data, neither HER2 or HER1 and 3 or TOPO2 were predictive of anthracycline benefit. They were prognostic, but they were not predictive. And this is a trial with 80% power to detect those interactions. Many of the previous studies, including the one we recently published ourselves on BR9601, in isolation, are clearly underpowered to detect these strong interactions, hence the need for the meta-analysis that Angelo described. But what does appear to be the case in 3 trials now, BR9601, MA5, and NEAT, is that polysomy of chromosome 17 does appear to be a marker that could be used to predict for anthracycline benefit. It's also an effect that was observed last year here in the N8931 study, and the fact that polysomy 17 is a surrogate, or rather TOPO2 is a surrogate for polysomy 17 also means that we might explain the BCRG006 results on the basis of this marker as well.